Sounds great. I'll keep a lookout for Lisa.
No worries. Is that working? Can somebody give me a high sign? Is this
We're
working?
good.
Yes, you're working, Jeffy.
Perfect,
That's great.
Jeff.
I'm going to blow through the first few slides. Thanks for inviting me. Thanks for Sarah, who couldn't be here. This is my conflict of interest disclosure. Yale has been awarded US and foreign patents and has filed provisional patents regarding some of the content I'm going to present today. All our work starts in the classroom, believe it or not. And this is a real classroom from the school not far from, just a few blocks from here. This is one of our intervention programs. And it's a great backdrop because it's real. It is one of our longitudinal studies on learning disabilities. Worldwide, about 15% of kids struggle with learning disabilities. They include reading disability, commonly known as dyslexia, language impairment, which is commonly known as delayed onset in speech. And now that you guys go to grad clinic, you know all about that. Verbal trait disorder, which is a problem of articulation. Dyscalculia, hard to say, but present, it's math disability. Attention disorders, again, you guys know a ton about this, I'm sure now. And for the most part, not for the most part, all the kids I'm going to talk about today are kids in regular classes, at regular schools, and with normal or above normal IQs. They have a normal distribution of IQ. These are not special schools. These are really just New Haven Public Schools about some of the programs we're going to talk about today. When we talk about learning disabilities, they are caused by multiple genetic variants, or they're polygenic. You have to be lucky enough to inherit a number of them, and they interact. And all of the learning disabilities, especially the 5 I've listed here, are highly heritable traits. That is, that the majority of the phenotype you're seeing in every individual, not some individuals, are caused by the action of these multiple genetic variants and interaction with the environment. But the genetic piece is actually often, usually higher than the environmental piece. The nice thing is that for some children, there is a durable intervention that can make a huge difference. And all this predated my work. This is data actually from Maureen Lovett. Maureen is a colleague at the University of Toronto in sick kids up in Toronto, Canada. Maureen's been working on intervention for reading disability for 30 years. She has NIH-sponsored randomized controlled trials. And what she's showing here in this graph, this stylized graph of her work over 30 years, is that if you identify children early here in grade 1, 2, or 3 that are struggling with reading, and you offer them a really intense intervention and then test them 2 years later after completion of the intervention, about 75% of these kids will be reading at grade level. But if you miss the diagnosis and you only pick up the kids up here when they're in 10th, 11th, or 12th grade, only about a third, if you're lucky, of those kids will respond to even an intense year-long intervention. So to identify kids at risk for dyslexia at an age when intervention works best, shown by Maureen and other people as well, we developed a genetic screening panel based on saliva. And so we never go after any of our subjects with a needle, it's always with saliva. And from that, we can extract DNA. And from all the studies I'm going to talk to you about today are from that extracted DNA. So we developed a screening panel by doing a bunch of different genetic sort of study, genetic association, genetic linkage, analysis, deep sequencing, rare variants. In other words, we'll do anything to try and extract the data, the information that we need. And then, of course, we publish them as best we can. Probably have over 90 publications at this point on most of this stuff. We use a range of approaches. So obviously in the classroom, we use neuropsychological approaches, and I'll show you some of the batteries that we use. And then from, as I mentioned, we can extract information from actually the DNA. The idea being here is that since the heritability is so high, probably the best way to find these kids at the lower end, since testing doesn't really work very well, is through genetic analysis or genetic screening. So what I'd like to do is show you our signature project, which we call the New Haven Lexinome Project, which we're continuing to analyze, even though we concluded enrollment about a year ago. The goal of New Haven Lexinome Project is to explore the extent to which genetic variants correspond with response to intervention through whole genome sequencing, serial behavioral testing, really not neuropsychometric testing, and serial functional MRI studies. It's a longitudinal study. These are normally developing children, grades 1 through 5. We recruited children from over 30 New Haven public schools scattered around the county. We've enrolled so far about 500 children, and we have extensive parent questionnaires that explores past medical history, socioeconomic status, learning disability history, et cetera, et cetera. And for each one of these 500 children, we have 30x. That's very deep whole genome sequencing. We also do standard psychometric batteries where we assess reading, language, executive function, a little math, a little rhythm. We do these serially, or did do them serially from grade 1 through grade 5. So let me show you a little bit about how the data works and how we do the analysis. And what I'm presenting to you are subtests from 3 standardized batteries, right? This is the test of word reading efficiency, known as the Tower. This is the Woodcock-Johnson. This is the Gort. I didn't invent any of these. These preceded me, most of them by 25 or 30 years. They've been normed, embedded on literally tens of thousands of children in the United States and in Europe, Australia, et cetera. From these, we combined about 5 different subtests, sight word efficiency, et cetera, into what we call a composite. In this case, we're calling it a decoding composite. Basically, we average the results of these. So let me show you how this works, because I think it's a bit of mystery how you connect genetics with the psychometric testing. And it's really quite simple. Let's say we use a single nucleotide polymorphism, and everybody knows here in this group that a single nucleotide polymorphism, or SNP, is basically a single nucleotide that's mapped to a single location. So it occurs once and once only in the 3 billion bases of the human genome. And that single nucleotide polymorphism generally has 3 flavors. It's either homozygous for the 1 allele, or homozygous for the 2nd allele, or you're heterozygous in the middle. And the idea here is you just basically do a linear regression based on performance for that individual, for that single SNP. And so here the idea is, in my stylized representation, is that if, let's say, low performance here on decoding is, at the bottom, and on the y-axis, high performing would be here. And so if if you have 2 copies of the A allele and you're a poor performer, and then sort of in the middle with only 1 copy, but you're a good performer with GG, then you can see here is you're making a linear correlation between this SNP and actually performance on this particular phenotype. And that's how the game is played. You could interpret it another way. You could say, oh, it's GG because it's protective, but regardless, that's kind of how the game is played. So I presented to you basically 1 SNP, but in fact, we can pretend it's on chromosome 8, but in fact, you can do literally hundreds or thousands of SNPs on chromosome 8 that span the entire chromosome, as well as chromosome 7, 6, in fact, all 22 autosomes and the 2 sex chromosomes. And that's represented here by what's called typically a Manhattan plot, which you've seen before and probably never knew what it was called, but you can see obviously why it's called that. The black and gray are just used to separate the chromosomes. Notice here there are more SNPs on chromosome 1 compared to, say, chromosome 22, and that's because chromosome 1 is the longest chromosome, so it has the most number of SNPs. The higher you go on this y-axis, which is the minus log 10p, represents really a smaller p-value, so higher confidence. And what you can see is, actually, this is real data from our cohort, is that we're beginning to get a signal here, very unique signal stacked up by multiple different SNPs at a chromosome 19. And so this is, when you look across the genome, this is what people call a genome-wide or association study, or a GWAS, because you do exactly what I just showed you, the single SNP, literally 750,000 times for each of the 500 subjects. That's the game. So here we really did get a signal. So our decoding GWAS shows a peak of SNPs achieving statistical significance on chromosome 19, even after we correct for sex, age, race, ethnicity, socioeconomic status. Basically everything we tried to do to make sure that this isn't a false positive, it seems to survive. It's very nice that it's very sort of single and all in one location, but let's get a high resolution. In other words, what are we hitting here? What are the genes that are encoded here? And so we can do that with our little magnifying glass, and there they are. This is a little segment from chromosome 19 showing 4 different genes and all our little SNPs with these weird names, RS whatever, RS whatever. The red guy here would correspond probably to the highest guy here. Now, before you ask me, I'll tell you none of them are encoding region, and that's typical for learning disabilities. Remember, these are children with normal IQ, so there's no major disruptions of any of these genes. But you can see most of the SNPs are falling out on chromosome, I'm sorry, on a single gene, which we had never heard of before called Gary1. So we thought that was pretty cool. This is a pretty good candidate. Let's take a look at it. And so we can do some biostatistical analysis. One of the things that we can do is look at GenoSkyline. GenoSkyline helps identify epigenetic regulatory regions, and you can do it by organ system. In this case, we're looking at the brain, which we think is probably pretty important to reading. And so when we look at the brain, multiple areas of the brain, in fact, there is one of these epigenetic regulatory regions in this particular gene called Gary1, which we identify by GWAS, and they happen to fall on regions of the brain, the cingulate gyrus, the inferior temporal lobe, angular gyrus, which are critically important to reading. So good evidence that we're probably in the right place. This is really the take-home data, because what we can now do is go back into our cohort, and we can ask the question, what happens to performance if you have one copy of the deleterious allele from this gene that we identified by GWAS? And we can look, now that we have all this formal neuropsychological testing multiple times, we can look at any of them in all our kids, and we can identify kids that have one copy here in orange of the deleterious allele and compare them to kids in the same cohort who have 0 copies of it. And no matter what we look at, word letter ID, word attack, which is a timed reading, single word reading, fluency, but what most people really care about is comprehension, is that you can see is that, and we look at the same kids over and over, start of grade 1, end of grade 1, start of grade 2, end of grade 2, all the way to the start of grade 5, is that coming into school, you're performing more poorly compared to your peers. And even all the way through 5th grade, these error bars don't cross. You're still not performing what your other peers are doing. So this has a pretty significant effect. And it affects multiple different subtests, or multiple different modes of ways of assessing reading. Well, we can go back again, do another bioinformatic analysis, which we did. One of the most prominent ones, or interesting ones, is this GTEx, where we look at expression levels throughout multiple organ systems. It's publicly accessible, just like GenoSkyline. And yes, Gary1 is expressed in the brain, but it's really expressed in cerebellum, which is sort of interesting because it brings up this idea that was first put forward by Nicholson and Fawcett back in the 90s, called the cerebellar theory of reading disability. And what they observed is that reading disability has been associated with deficits in cerebellar related functions like dystonia, time estimation, skill automatization, which I can't say, and balance. And so what they concluded was the cortical cerebellar circuit, that's this guy here, seems to have an important role in reading. And I can tell you it's changed the direction of some of our research projects. We're exploring this in depth right now in several large data sets.
Hey, Jeff, 2 slides ago, although they started out behind if they're missing one copy, but were you surprised that the slope and the amount that they were behind didn't change over time?
Shocked. Yeah, okay. Shocked.
I would have thought they would have started out lower and not have gained as much during school.
They don't catch up.
But nor do they fall further behind, is what I find interesting.
Oh no, well, remember these are smart kids, and while many people don't recognize it, the fact is that they're in good schools and they're getting good education and with good teachers. So as best they can, they're trying to keep up. Okay, so that's the first part. Next question is, okay, cool. We can do this genetic thing, and we can find these genes, we can find these variants. What's going on in the brain? So since we have the imaging data from the Ed Project, plus we can plug into other data sets, we can do actually an imaging genetic analysis where we predicate our genetic analysis now, not on performance, but actually on brain MRI data, either structural or functional, which is a really cool thing. And I won't bore you too much with this because we published this back in 2018. But for example, we can look at, this is a regulatory region called READ1, or a regulatory element called READ1, which regulates expression of a reading associated gene. And we can look at that in particular reading well-known, well-described, not by me, but by others, reading circuit in our kids or in any large data set where there's functional data. And then again, we can do the same sort of approach, a linear regression, and look at the relationship between two different nodes within the reading circuit. And what you'll see here is the right superior middle gyrus and the right inferior frontal gyrus or the insula are actually highly correlated with whether you have one or the other READ1 genotypes. And so this is what was called imaging genetic analysis for obvious reasons. And here it's predicated on an interesting brain function. Okay, that's cool. What about on a cellular level? Can we look at it in neurons? Well, we know a little bit about brain development. And as I didn't emphasize before, but we have the data, is that all these genes that I've been, or certainly the one I've been talking about so far, Gary1, and some other, and READ1, they're all important during neural development early on when the brain is developing, mostly at the end of the first trimester. Well, we know a little bit about neuron or neuronal development. We know it begins with embryonic stem cells deep in the periventricular space. They evolve into, or they differentiate into neuroepithelial cells, and then the program starts in neural progenitor cells, intermediate progenitor cells, et cetera, finally to mature neurons. And while this is happening, so is neuronal migration. The cool thing is that there are immunofluorescent markers that are the hallmark of each of these stages. And so nanog for the embryonic stem cell, neuroepithelial cells, et cetera, et cetera. So if we could go into kids' brains, we could actually begin to look at this and look at some of these genes. But most of you know, if I were to ask you for a small little biopsy of your child's brain, probably wouldn't be too thrilled about that. So it's the next best thing. And so we use human embryonic stem cells and we differentiate them along a neuronal lineage. So now we can manipulate them using the magic of CRISPR. And in this case, we have a lentivirus that targets a well-known reading disability gene or dyslexia gene called KIAA0319. And then using CRISPR, and which within an embryonic stem cell line that's expressing DCAS9, we can actually knock down expression of this well-known reading gene and see how it affects neuronal development. And what's really interesting, and we can do this initially by looking at our immunofluorescent markers, is that even with knockdown of the KIAA0319, and it is knocked down, what we see is normal progression to an expression of SOX10 in neuroepithelial cells, but then accelerates out of neuroepithelial cells into neural progenitor cells. In other words, they slow down expression of SOX10 and they increase PAX6 faster in a shorter timeline than the controls. And then they stop. They go into developmental arrest. Cool. So what we then did was we sampled RNA from at an interval of 1 week intervals, day 7, day 14, day 21, day 28. We looked at knockdowns compared to controls. We looked at overexpressed versus underexpressed genes at each of these stages. And when you do a study like that, what you can do is you can do a pathway analysis. And when you do that, something very interesting came up. There's a number of pathways that are involved. And what they implicate is that cells which would normally go without a knockdown from neuroepithelial to neuronal progenitor, then to an intermediate progenitor, which would be the active, they actually become quiescent. So KIAA0319, a gene identified by clinical studies in the way that I showed you before, the GWAS thing, actually identified by another group, not our group, favors entry of neuroprogenitor cells into the quiescent state. And I thought that was really, really interesting. I'm sorry, the knockdown favors entry. So without the knockdown, you go through it, you go through the normal progression. But what KIAS1 seems to be doing is it's pushing or maintaining this normal neuronal differentiation progression. Now, whether it goes through the active state or goes straight into an intermediate progenitor under the influence of KIAS319, we don't know, but that's what the next year is going to tell us. Okay, so we've done the neuroscience thing, we've done the micro thing, right, the cellular thing. What about on a macro scale? What do we know about genes that affect reading and their possible influence on the evolution of the world's languages? And so to address this question, what we did was we looked at languages from different populations around the world. I'll give you an example. This is the Igbo population from Africa, and this is a Japanese population. And what we looked at were two things. We looked at a frequency of a regulatory feature called RU1-1 and compared it to the frequency of consonants in that particular language. So for example, in the Igbo language, they have a high percentage of consonants in their language, whereas because it's red, right? And the Japanese language is a light yellow, they have a lower. And then the size of the sphere being high for the Igbo, it means a high RU1-1 frequency, and the smaller sphere means a lower frequency. So high consonants, high frequency, low consonants, low frequency. But in fact, we didn't do just two populations, we did 65. And so when you look at this around the world, and then you account for geographical distances, in other words, obviously there's going to be genetic sharing, and people migrate one to the other. So if you account for all of that, then you can actually do, again, another linear regression. And this time, what we're doing is regressing the frequency of RU1 on consonants. And you can see here is that there's a really nice relationship. Interestingly, if you look at vowels, there is no relationship. So a nice sort of built-in control. So it's very interesting that the implications here of genetics and the evolution of human language. Okay, I'm going to do one last area, and that's an educational approach. So our current model in most school systems, private and public, around the country, is this idea of wait to fail. Well, let me show you what I mean. Let's say we do any one of our standard reading assessments, let's say in this case, comprehension. This would be a typical development of a child from grade 1, grade 2, grade 4 for comprehension. It's not exactly linear because nobody's, human behavior is never linear. This is pretty typical of what it would be. Relatively low starting, relatively high later. For kids with reading disability, they begin to fail usually late grade 1, grade 2, once the curriculum gets really challenging. That's when they really get into trouble. Before they can really fake it, especially if they're smart, this is when they really get into trouble. Standard intervention would be for them to wait till they fail, and then at some point later, do an intervention. And very similar to what I showed you with Carry One, they never really catch up. There's always this gap. Our idea would be some sort of early identification, right? So that we could do early intervention, get them up to grade level or very close. And we would call this, say, if it's based on, say, genetic screening, precision intervention. But to do this, we have to get out of our ivory tower and somehow convince school systems and legislatures that genetic screening is a viable thing and a worthwhile thing to do to identify these kids early. So I think I'm close to the end of my time. I won't be much longer, but what I tried to show you was that we generally take a multidisciplinary approach. Neuropsychology, biostatistics, education, neuroscience, genetics, and a microscale and a microscale and macroscale. And in our group, we really do blur the boundaries of all this. We have lots of people come in with a neuropsych background, know nothing about neuroscience, and they learn it. And same thing for genetics, they'll come with a genetics background, but they know a lot about neuropsych, but they learn it. And so people do, you have to be good in multiple different things. And thankfully we have a nice depth and breadth of people to be able to do this. The work started obviously in the Department of Pediatrics and Genetics, and I already showed you some of the stuff we do in computational biology. But if we really want to impact the world, we really got to go beyond this. And so what we've begun to do this year is we're doing a course in the Yale Educational Studies programs to begin to educate undergraduates that are interested in becoming teachers here at Yale. And the other thing is that there's the new Broad Center for Education at the School of Management, where they bring in administrators from school systems around the country. And so we have access to them too. So the idea is spreading the word. And so we're using all this integration and our efforts to try and spread the word really to build this brand new Yale program for learning disability research, which we launched just this year. So let me summarize what I've been doing. I tried to tell you guys, what I tried to do is present some of the highlights of our work over the past 20 years to identify what I think is a significant problem and to implement sustainable change. But I don't really expect most people to remember, you know, GWASs, linear regressions, DCAS9 knockdowns, et cetera. So here's my take-home message to the fellows, since that's what Sarah asked me to talk to. So fellows, if you're online here, this is what I'm trying to tell you. These are my take-home messages. Because as you navigate your careers, you encounter problems, and I hope you begin to think about how do you implement durable change in some of the problems that you're identifying. So this is what our approach has been. Number one is we figure out what the problem is. What's the fundamental question? What are you trying to address? That's critical. Discern the underlying mechanisms. In our approach, we don't care. We don't care if it's I'm not trained in neuropsychology. I'm not trained in biostats. I'm barely trained in neurogenetics. I'm certainly not trained in neuroimaging, but we use all these approaches because they work and we can find expertise to do this. We then try and understand the barriers to implementation. And we don't have time to talk about this today, but many of them can be sociological, they're financial and structural, but you have to identify them first so you know have come up with some sort of plan to address them. Devise strategies to overcome the barriers. In our case, we think it's genetic screening. I think in older kids that aren't going to really respond to early intervention, I think eventually some of our cellular stuff will lead to potential drugs. And I think that could really be interesting. But if you really want to make a durable change, you have to think about local policy, state policy, and federal policy. And that's why we really built the Yale program for learning disability research, and why we're reaching out to School of Management, why we reach out to other advocacy groups around the state and around the country to do this. And then lastly, it takes a fearless pursuit of the goal, regardless of the challenges, whether they be financial or methodological or attitudes or prevailing sentiments in the community, just push through. So that's my 20 minutes. Here's a small portion of the gazillion people that helped to do this work. Big shout out to Sarah, and thank you all. I hope that was helpful.
Thank you, Jeff. That was great. That was awesome.
You're welcome. Did Lisa ever make it?
Lisa's on. Lisa's on.
Great.
I'm here.
Can I ask Jeff one question? Jeff, can I ask you one question? You know, when you were showing your one KIA gene that you're talking about, like, leads to quiescence. And, you know, the genes and the pathways that lead to quiescence, like even in single cell organisms, are conserved all the way to people, like mTOR pathways and autophagy, et cetera. So you're saying that this is happening in brain development also? Is that can I remember that as that's true? Or
you know, the whole idea of using human embryonic stem cells to recapitulate what's going on in the development brain is sort of a new field. What we've done is we've done 2D cultures. We've now done organoids. And we haven't done the knockdown yet in organoids, but that's going to be the next step. And so it depends, you know, slightly on where the field is. I mean, our work can't be beyond where the field is. So I think the $64,000 question is, to what extent does 2D cultures, human embryonic stem cells that are enriched for neurons and even organoids, how do they really recapitulate normal human brain development? Yes. Are we showing something that's really unique, something that wasn't shown before? Yes. Is it really interesting? Yeah, it blew my mind. But is that really what's going on in the brain? I think so, but I don't think we're going to know until as the field moves forward and finds out, well, what is the relationship between these cellular systems and normal human development? And I think people are starting to do that, and so we'll see what happens. Great question.
And a lot of that is related to environmental cues. A lot of quiescence. But so, is it a culture thing, or, can you model it some other way?
So we're just saying it's pre-programmed. It's there. So, and maybe the default pathway.
It's great.